Hello and welcome to channel 87,592, The Dream of a Labean. I'm your host, Carl the Bean Johnson. Today we will also have Billy Noodle on the show, followed by our tech expert, Baby Pubble. He's not currently in the show right now because he's working on our cameras and stuff. Today we have an interesting idea that we want to explain, and that is DNA replication. An interesting topic, but there's a lot to learn, and there's most like a lot of things you don't know about it. Let me hand it over to Billy Noodle to explain the first couple ideas. Hey, can you fix this baby? I'm trying to play the video. Alright, got a couple seconds.
An amazing sight to see as we zoom into the origin of living things, the cell. Zooming in a little further, you can see a sphere shape inside the cell, which is the nucleus. It's the place where most events of replication will be taking place. Even further, you can see structures that look like extended X's called chromosomes. These are responsible for individual genes.
Wait a minute, I'm wearing jeans.
That's not the right gene.
Oh, okay. Sorry, I want to make sure.
Anyways, after the cliche moment there, the chromosomes are responsible for our genetic makeup.
Makeup?
Is there any chance you can shut your mouth for a minute?
Sorry, I'm just a little confused because I lost my script.
And you're the host?
Yep.
So, I was just saying, chromosomes are responsible for our genes, which make us up. Eye color, hair color, and many other things about us are impacted by genetic makeup. Now, returning to our video, you can see that the chromatids are coiled up in the chromosome. These chromatids consist of deoxyribonic nucleic acid, or what I like to call it, DNA. DNA is like a twisted ladder many of us call a double helix. These have 4 nitrogen bases, A, T, C, and G.
Oh, I know this.
Take it away then.
Alright. A stands for adenine, T for thymine, C stands for cytosine, and G stands for guanine. U stands...
Wait. U comes later. It's not a chemical in DNA. It's only an RNA.
Oh, okay. Anyways, these nitrogen bases come in bonds. A binds with one side of the ladder with T, and C on the one side of the ladder binds with G. These can be arranged in any order. However, DNA A always has to bind with T, C always has to bind with T. This is called...
complementary base pairing?
Yeah. The way I remember it is that apes are to trees as cows are to grass.
What?
Anyways. Oh crap, we went overtime. I guess it's time for some advertisements like any good TV shows, which is the reason many are switching to... Have you had those days where you're just kind of down? You're like, why? Why is a pizza $15? Well, I have a solution for you. I went to McDonald's today. All this and more, $5. Beat that. For McDonald's. After all, I'm loving it. Are you having trouble getting your clothes clean on a regular basis? Have you tried all the brands? Have you tried, let me think. Well, there's Mr. Clean. There's Dawn. There's other cleaners. But nothing can be as clean as this. It's a Tide ad. Hello and welcome back to our DNA show. It's time to get into the replication part of the DNA. Hey Billy, are you doing alright? Oh no, not another one. Hello and welcome back to our DNA show. Now it's time for the main part, the replication of DNA. Fortunately, Billy Noodle has left because of a heart attack, so I've called in our tech expert, Baby Bubble. Nothing to worry.
Well, thanks for having me on the show today, Carl. Let's get back into the video as I read off of Billy's script. Let's see here. So now an enzyme called helicase appears and it splits the DNA, not all the DNA, but a portion of it.
Yeah, like when a zipper gets stuck and the middle is unzipped.
That's a good analogy. Well, now pieces of the RNA come together and bond to the unzipped selected side. This is basically the same thing as DNA, but there are some differences. For starters, the A with T and C with G rule is slightly changed. Instead of using T, thymine, RNA uses U, uracil.
Oh, so that's what the U comes in at.
Another thing is that since RNA is a copy, there is only one side of the ladder, or a single helix. RNA also comes in more than one type. There's a messenger RNA, which is a copy of the DNA, and there's a transfer RNA, which is, well, I'll get into that a little bit later. Another thing is that's cool about RNA is that it can actually pass through the nucleus and enter the cytoplasm.
Right, yeah, the cytoplasm is like that goo or that, space between organelles.
Yeah, anyways. Now I'm going to get into the real meat, if you will, of the DNA replication and the linking of proteins. Now we start with the ribosome. The ribosome is the machine, if you will, in the next process. Now next we go to the transfer RNA and the tRNA. This appears in the clover-esque shape and is responsible for the code of protein linking. Basically now the copied RNA strand from the DNA or transfer RNA will be divided, if you will, into pairs of 3 nitrogen bases. The set of 3 bases on the mRNA is called the codon, and the set of 3 bases on the tRNA is called an anticodon. Sort of like DNA, these will pair up, but since the RNA, A will bind with U, C and G are not affected and pair together just like normal DNA. Anyways, this process repeats again and again as the proteins from the last tRNA build with the proteins with the tRNA. This process will eventually stop only until a codon reads stop. You can see a list here of all the different proteins that different setups of RNA will code for. Now that you have your protein bonds, it can go further into the cell and perhaps sent on a journey to other parts of the body. Carl?
Oh, sorry. I got lost when you said ribosome, and from there on. Oh. Well, it looks like it's about time to wrap up our session for our show on channel 87,592, The Dream Of-A-Labean. Thank you for tuning in today and listening to our special talk about DNA and DNA replication. Next week we'll be talking about the crippling long-term effects of McDonald's. We'll have Barbara Chubb and Stephen Biggie as our guests next week. Thank you, and once again, I hope you learned something today. One thing I've learned today is to remember my script and not to freak out when a colleague falls over for the 900th time. See you later. This is Carl Bean, signing off.